From an idea formed in the mid-90s and from the advanced study of the relationship between regenerative medicine and quantum physics, a revolutionary system for applying stem cells on humans, providing exceptional results without side effects or legal restrictions, came into being. This proposes itself as the most economically interesting project of the last 50 years. The key to using stem cells and placing them on the market. For the last 15 years, the aim of traditional research has been, and still is, that of finding stem cells with characteristics that make them eligible for human trials, valid for the treatment and prevention of many illnesses and without side effects, a type that has still not been found. Up to now, embryo stem cells that are pluripotent have been tried, but this poses ethical questions, and they are not suitable for clinical use, and they cause tumors. Even induced pluripotent stem cells, for which Yamanaka has been awarded a Nobel Prize, whilst they are extremely useful in vitro for the study of adult stem cells, they cannot be used clinically because of their intrinsic capacity to proliferate, thereby forming teratoma. Adult mesenchymal stem cells obtained from bone marrow and fat do not have the value of regenerative pluripotent stem cells. In fact, they are only able to transform themselves into some tissues. They cannot be used in human therapy because a few days after being put into culture, they begin to differentiate themselves and show different receptors on the membrane. Therefore, they become a heterogeneous cell population. For this, they cannot be used in human trials because by law one cannot experiment with something that is not qualified and quantified. The same applies to bone marrow and fat stem cells obtained by concentration, not culture, because they are mixed with other cell populations and debris, therefore they cannot be qualified and quantified. Stem cells obtained from blood through apheresis are hemopoietic and therefore can only be used to treat blood illnesses. Even if these cells are not pure, legally they are autologous blood derivatives and have therefore been accepted for curing illnesses such as leukemia. Stem cells obtained from the blood of the umbilical cord have the same hemopoietic characteristics as those obtained through apheresis, but they have the histocompatibility factors that identify them with the donor, so in order to use them for curing blood disease, it is necessary to obtain from a bank stem cells having compatible characteristics with that of the patient. All these kinds of stem cells share the same complex production process, the use of embryos, transferring pluripotent factors through a virus into colonies of adult stem cells and maintaining them stable, taking a bone marrow sample from the sternum or a bone, taking a fat sample from subcutaneous tissue, putting the cells in culture for many days, organize umbilical cord banks and database to evaluate compatibility, etc. The type of stem cells obtained through our patented system allows us to overcome all these difficulties. From a simple blood sample of a few mLs in 72 hours, we obtain a vast number of hemopoietic, mesenchymal, pluripotent adult stem cells, all having only stem cell factors, therefore qualified, pure and at the same maturity level. We can separate and count the three cell types into the percentage required by our therapeutic protocol through sorting process, therefore quantified. Moreover, our stem cells show no histocompatibility factors, that is, cells from another individual can be used on a patient without the risk of a reaction. This means that besides an autotransplant, it is possible to create a real drug, that is, one could produce thousands of doses from half a liter of blood from a single donor. Experiments on non-provoked pathologies in horses and dogs, animals near to man on the zoological scale, have led to a safe therapeutic protocol. The inoculations carried out so far in veterinary are around 15,000 autologous and more than 5,000 allogenic, that is, from another individual, without side effects when injected following our protocol. The fact that stem cells obtained through our patented system are hemoderivatives and show only stem cell receptors makes them eligible for trials in human medicine. Conclusions. This is the only real possibility of using stem cells for the prevention, treatment and cure of illnesses and to slow down aging by optimizing the psycho-neuroendocrine immune system. The stem cells obtained are not a therapeutic protocol, but work within every therapeutic protocol alongside drugs and surgery. They are obtained from autotransplant and also from donors because they do not have a passport. They take on the passport of the patient and have the typology of a real drug. In the future, there will be no clinic or hospital that does not have stem cells ready for every emergency pathology, such as heart attack or stroke. These cells must be substituted every 15-20 days. The percentage of pluripotent stem cells obtained through our procedure can improve many types of neurological pathologies that are at present incurable. We are facing a market that will require within 10 years more than 500 million doses annually, with the tendency to grow because these cells can be used for prevention and within every therapeutic protocol.